welcome back to Big Things Made Simple. Today we're going to be talking about mitosis. So before we get into the actual mitosis process, let's go through some key concepts and info that will help us understand mitosis better. At the base of mitosis is DNA, aka deoxyribonucleic acids. In non-dividing eukaryotic cells, DNA is found in the form of chromatin and is loosely coiled and wound around histone proteins. In dividing eukaryotic cells, however, the DNA is more tightly coiled and denser and can be seen as chromosomes, which are visible under a microscope. These chromosomes, prior to DNA replication, will exist as a single thread, known as a chromatid. However, after DNA replication, these chromosomes will contain two sister chromatids. They are named sister chromatids as they have genetically identical alleles. The sister chromatids are joined together by a centromere. The centromere is also the site of attachment of spindle fibers. It is important to note that the DNA at the centromere has a short sequence of bases that repeats many times. However, it is only used to maintain structure and does not carry genetic information. So what are genes? Genes are basically sections of DNA that contain coded information in the form of base sequences. A DNA molecule has many genes along its length, and each of these genes will occupy a specific location. The more technical term for this is called locus. These genes can also exist in many forms that are known as alleles. Since alleles have slightly different base sequences, they will code for a different polypeptide and so a different protein. New alleles of genes can be produced by mutation, as mutations are changes in the base sequence of an organism's DNA. Now let's take a look at poly level. Poly level refers to the number of sets of homologous chromosomes in the genome of a cell or an organism. The poly level varies from species to species. Some will have diploid cells, others will have triploid, tetraploid, hexaploid, octoploid, and so on. Of course, with so many different species in the world, it is impossible for us to remember each species' poly level. But thank goodness, we only need to remember humans. So in human body cells, we have a diploid poly level. In human gametes, we have a haploid poly level. In conjunction to the poly level, we have chromosome numbers. Different species will also have different numbers of chromosomes in each set. So in humans, haploid gamete cells have 23 chromosomes, and since chromosomes exist in homologous pairs, the diploid human cells will have 46 chromosomes. The chromosomes in the homologous pair are identical in size and shape. They carry genes for the same characteristics at the same loci, which is the plural for locus. They are not genetically identical, as one chromosome of each pair comes from each parent and therefore will have different alleles. Hence, they are not genetically identical. Some pairs of sex chromosomes, such as the X and Y in male mammals, are different sizes and are therefore not homologous pairs. Moving on to the human karyotype. Karyotype meaning the number and visual appearance of the chromosomes in the cell nuclei of an organism or species. Now there are two classes of chromosomes that we need to know. Autosomes, which are chromosomes that are not involved in determining sex, and heterosomes, which is the X and Y chromosomes involved in determining sex. Now that we know all the basic information needed to understand mitosis, let's begin. For a cell to undergo mitosis, it must go through the cell cycle. The cell cycle is basically a series of events that takes place in a cell, leading to its division and replication of its DNA to produce daughter cells. The cell cycle can be divided into three main stages: interphase, mitosis, and cytokinesis. The length of cell cycle varies in different tissues. For example, root tip cells of onions divide every 20 hours, while the epithelial cells in human gut divides every 10 hours. So let's take a look at the first stage of the cell cycle, interphase. Interphase is known as the longest stage in the cell cycle. Here, the cells are very metabolically active as it is synthesizing new molecules and organelles and carrying out DNA replication. In interphase itself, it has 3 phases. The first phase is G1 phase, G standing for growth. In the G1 phase, proteins are produced to synthesize cell organelles, nucleotides, and histone proteins, so that when the cell divides into its 2 daughter cells, the daughter cells will have an equal amount of organelles, histone proteins, and nucleotides that is needed for its function. And because these proteins are being produced, the cell will double in size to accommodate the increasing amount of proteins. Here, there are 2 centrosomes present, and each centrosome will only contain 1 central. The second phase is S phase, which is also known as the synthesis phase. There are 2 things that happen here. First, DNA replication. The DNA will be unraveled and replicated, and this newly formed DNA will then combine with the newly produced histone proteins that were produced in the G1 phase, hence the amount of chromatin in the cell will double. The second thing that happens here is the replication of centrioles. So each centrosome will now contain 2 centrioles. The third and final stage of the interphase is called G2 phase. Here, the organelles will grow and divide, so that the number of organelles will double. Proteins are synthesized here as well, such as histones and enzymes. Energy stores are also increased in the form of ATP. And lastly, specialist proteins called tubulins are also synthesized here. They are used to make spindle fibers, which will separate the chromosomes in the later mitosis stage.